turn my camera. I wish I knew where it was. I might use it in the future. Okay, change to slideshow.
It's not sharing. Oh, here we go. Everybody can see that?
Yep.
Did you get my question, Jen?
Um,
I sent this morning.
I did not.
It's by email.
Oh, okay.
Yeah, they are big.
So we do have some attendees already. We're just going to give it a few minutes for others to join before we get started.
Sure.
My phone on mute.
Advances? Yeah, good. Yeah, 12 participants.
Yeah. So again, thank you for those who have joined so far. We're just waiting a few minutes for others to join as well before we get started.
Jen, we should get going.
Okay.
Yep.
All right, we'll get going. I just want to welcome all of our attendees to LabShire's Ask the Expert series, an introduction to measurement uncertainty and how it can impact your laboratory operations. My name is Jen Bardsley, and I'm serving as the moderator for today's webinar. And during our time together, our panelists will provide their experience and insights on an introduction to metrology. During the presentation, please feel free to submit questions using the Q&A button on the control panel. And you can also, if you have any questions about a particular slide, you can make note of what slide number that you have a question on, and we will have a Q&A session at the end of the presentation. Now, I would like to begin our presentation by introducing you to Dr. James Blackwell. Dr. Blackwell is president and principal consultant of the Windshire Group and Labshire, which he's been a leading industry consultant for more than 15 years. Founded in 2011, the Windshire Group accelerates development and manufacturing for commercialization therapies and providing quality expertise. And Windshire has a new spin-off company called Labshire, which is a global laboratory consultancy that delivers world-class expertise in all aspects of R&D and compliant laboratories. Now it's my pleasure to turn the presentation over to Dr. Blackwell.
Thanks, Jen. I'd like to introduce Mark Gusak and Deborah Miller. They're world experts in laboratory operations, and you can see some of her background here. Mark also has extensive experience also in laboratory operations, particularly as a pathologist, and has operated and managed very large laboratories. They both provide their consulting expertise now to the industry. With that, Mark and Deborah, you have the privilege of hearing from two of the world's experts on this today. So pay attention and ask questions at the end. Thank you.
So I think our expertise partially comes from life experience and learning what went wrong and how to do it better. In this case, we're looking at what does good look like? What does it look like to the instrument vendor when they say the test results look good? And what does it say when they say the instrument is yielding good results? Good seems simple, however, it's actually the tip of an iceberg of a lot of information and data. We hope to reveal that iceberg to you today to help understand what good might mean to the instrument system assay vendor and what it must mean to the laboratory.
So anyway, my name is Mark Gusak, and I'm going to be doing most of the talking. But as I do this, Deborah will be coming in and talking as well. And so this is the first in a series of presentations where we're going to examine what good means. And in particular, to do this, we're going to introduce the following. The concept of metrology, otherwise known as measurement uncertainty, and how that impacts really everything that you do in the laboratory, whether it's a clinical laboratory, a research laboratory, or even a manufacturing laboratory that tests a drug product as it's going through the different points in the manufacturing process. And so basically, it bears upon your laboratory operations. And that's not just simply the routine operations. It also has to do with how you choose a laboratory testing system, how you have it installed, and how it's qualified for its performance. And then it's how it operates in the long term and making sure that it's producing the appropriate results for the end user. And that's the big deal here, is it impacts those who use the test results that you report. They're going to make decisions. In the clinical side, diagnostic decisions, decisions to change treatment or implement treatment or not. And that can be really critical in terms of the reliability of the testing. In terms of research, whether or not there is or is not a result that means that a new drug is worth testing clinically. And then, of course, in terms of the clinical test, if you're doing an operation that requires that the patients in the study be studied, that their blood tests are done, things like that. So it's not enough to say your test system meets vendor standards. And that's what the vendors are saying. Oh, that's good. It meets our standards. Because whether it's in the package inserts or it's in their qualifying protocols, that's really what they've decided based on the needs they have for everything from manufacturing to getting it through the FDA, as well as meeting the needs of a large number of clients, not just you. It doesn't necessarily mean that it meets your standards. So you need to do more than this, and that includes at least the following. You need to establish what the characteristics are of the test system as it operates in your laboratory, not in their development facility, okay? Not what they did to get the FDA to approve it. That's what's really important here. And then you need to establish acceptable performance targets for the installed test system. That is that meet the needs of your laboratory and your end users, not necessarily what they felt that they needed to do. And then you need to qualify the test system against those targets before testing starts, and make sure that it actually meets those, and not just what the vendor says they should be. And then once you get up and running, over the long run, you have to monitor that test system to assure that it doesn't vary randomly, so much that you know individual decisions really can't be reliably taken for patients or for the outcome of research, or drift significantly from the following, which is what we call acceptable performance, and that's based on acceptable risk criteria, which we'll be talking about more in the near future here. And I'm not sure why this is happening, but some of the I apologize, some of the animation has changed over the system. So Deborah and I like to talk about the mission of the laboratory. We feel that if you start with the mission, then go through what the purpose is of any particular test system in terms of meeting the requirements of your end user, the person who receives, or the persons who receive the test results, you're going to minimize the number of errors that you're going to make in choosing and qualifying and running during routine operations, and you'll get the best results for them, for their decision making. So the test systems for the presence and testing specimen for the presence and amount of one or more analytes is really what the mission is. And then providing these test results as reliable as they can be, given the limitations that we'll see going on in these test systems. And these include, but are not limited to, meaning that we're talking about just the tip of the iceberg, not the whole iceberg, which we'll get to in later presentations. So the test system used, either vendor provided or laboratory design, and this is important. I have inspected for ISO 15189 a number of large laboratories during the COVID pandemic, where they found that a vendor provided instrument did not meet their needs, and they actually had to design their own laboratory systems. And that really made things quite complicated and imposed certain limitations that vendors are able to get rid of. The systems in place to monitor the reliability of the test system itself. And that includes the personnel, the reagents, calibrators, controls, built-in control systems. Some of the newer test systems, such as for PCR, actually have their quality control built in. It's kind of out of the reach of the average technologist. And then the processes used to run the test system in the laboratory. So in research, they will be very, very controlled under the management of people and technologists who develop the system. They know it intimately, whereas in the laboratory or in the research lab or manufacturing lab, the processes are going to be written in part based on the vendor documents, but also are really going to be based on the technologists that are running the personnel and their qualifications that are running it. So it's important that these processes are designed to make sure that you minimize the metrological error.
Mark, a simple example of that is in a laboratory, some labs may run 3 tests at a time and others may batch it. And instrument performance can be different if you're running 3 controls and 3 samples, or if you're running 3 controls and a whole, like 24 samples. So
yes.
That would be a process difference.
Yeah, and then ultimately it's the people that matter. You know, yes, there is, of course, the amount of error that goes into the design and the manufacturing and the reagents, things like that, that are really not under the control of the lab. But what is under control is the competence, which includes the training, their educational level, how well they're managed, all personnel issues that can lead to problems with the operating system degenerating over time and producing unreliable results. So not all measurement uncertainty can be eliminated. And that means the lab needs to help the recipient of the results identify what is likely significant and what is likely not significant. And so in the clinical laboratory, it's really important to give the clinician an idea of how much variation can be attributable to the test, not to the patient, but to the test. And so they can better understand the patient's clinical condition. And an example of this is hemoglobin A1C for diabetics. In the VA, what they've done is for hemoglobin A1C on a high performance liquid chromatography instrument, they've actually determined approximately how much of the variation that is in the test system itself and not necessarily biological. And they now report this out with the test results so that a clinician doesn't suddenly change management in terms of what drugs are being used or how many are being used or what the dosage is on something that's probably just a statistical variation, a variation that is not biological. And so that they'll wait another 3 or 6 months because they don't want to over or under treat their patient. And the same thing is true in the research laboratory. You've got experimental systems that are being observed and or they're trying to control. And the whole idea is to not give a result that would cause a decision to be made on either way, to accept an outcome that does not give an indication that a drug is going to be good, or to reject a drug because the outcome seems to indicate that it is not. So you could err on both sides, and then a lot of money could be lost, a lot of time and effort. And so this means the laboratory needs to help the user of the test result decide what is acceptable measurement error. What's the acceptable risk here? And there can be risks in terms of liability, there can be quality issues, there can be financial issues, all of which they need to look at. And then that impact on their clinical or scientific decision making. So that's really the key here for why metrology is so important. And that includes things like FDA regulations to make sure that they're going to meet what the FDA states is necessary in order to approve the drug. So you can go through the whole thing. Clinical trials look great, everything looks great, and then the FDA comes in and says, well, we don't think that particular test you're using has the adequate accuracy or precision. Again, I'm not sure why this is happening. My apologies. So metrology, what is it? What's the definition? And what I like to do is to have as short and concise a definition as possible. So basically, it's a field that explores measurement uncertainty. And what we've come up with, Deborah and I, is like just really one sentence that kind of combines everything together from what we've seen in the literature and what we've talked about amongst ourselves. And so there's a component of measurement that can be attributed to error, either systematic, it's also known as bias, inaccuracy, drift. There are all sorts of different terms you're going to see in the literature, or random, and that's also known as imprecision. And that leads to a deviation of an observed value, that is a measured value, from the true value, and which is a value we can never know perfectly, but which we want to get as close to as possible, given the limitations of what we're doing. So all test systems that we're talking about at their heart, whether they're manual or automated, measure something. And so they're affected by measurement uncertainty. And even if it seems like it's a qualitative test, like somebody is looking into the microscope and they are going to make a diagnosis of something based on, say, histology. Yes, that seems qualitative, but actually in their mind, there is something going on there are signals going on in the brain, and there are limitations to how accurate those signals are, and therefore error occurs. So generally test systems measure an analyte of some sort, such as say sodium or an antibody or DNA or a drug, and it's in a specimen, which has a certain type of matrix. And it uses one or more methods applied through manual testing or automated instrumentation. So manual testing could be counting spores or colonies in a petri dish, or it could be counting the number of different types of white cells in a peripheral smear. And then automated testing, of course, can be a chemistry analyzer that uses any number of techniques. It could be mass spectroscopy, it could be high performance liquid chromatography, all of which are measuring an analyte. So that means that each test result represents a series of steps. And that starts with the development of the test system itself by the vendor or within your own laboratory. And it ends with the actual measurement of a specimen during routine operations. And so there's just a huge area of where numerous steps are involved, where there can be an error that leads to the potential for measurement uncertainty. And they can all add up together, or they can cancel each other out on occasion. But it's really important to understand this when you're thinking about providing a particular type of test for your client, and how you're going to actually choose, what methodology and what type of vendor and what particular instrument. All of that is really important. So each test can be impacted by numerous potential sources that are embedded in the system. And it's also embedded into the approach to qualification. So for example, if you don't study enough specimens during qualification, enough different specimens that will explore all aspects of the test system in terms of its linearity, in terms of different concentrations, in terms of perhaps interferences, then you're not going to have the kind of reliability that your end user is going to expect. And so that can impact the clinical or research activities. So it's necessary for the laboratorian to be well informed about the degree of error inherent in the various parts of the entire system, from design through manufacturing to installation, qualification, and into routine testing. Now, this is a big ticket. This is a lot of responsibility and a lot of work. But what it does in the end is it means that you're spending the least amount of money on the very best system that has the qualities and metrological characteristics that are needed for your end user. And also that will work in your lab, depending on it's a huge reference lab that has huge throughput, or is a small lab that just needs a small instrument to do the studies occasionally.
And all of this sets you up for the monitoring phase that comes after.
Yeah, and maintenance and just simply the complexity of the operation for the people that you're working with. So all this is facilitated in part by the test system vendors, and even if you do your own developed tests, laboratory developed tests, you're going to be buying off-the-shelf parts and pieces. That's been my experience. You don't actually manufacture something. You simply, instead of getting a complete unified system that's all integrated, you buy this part from that vendor and you buy this part from that vendor and you put them all together. And that's what one of my large reference labs did for the COVID-19. When they set up, they actually had, instead of one instrument, they had 5 instruments, but they were able to get the results they needed for the high throughput, which was 100,000 tests a day, which is almost unbelievable. And when I was expecting them, they were running almost 50,000 at that point.
And for
So
those that are operating CLIA labs, that a laboratory developed test requires you to essentially play the role of the vendor to develop the test. So that adds all of those processes that you have to do before you even get to qualifying the assay in your laboratory.
Yeah, exactly. So the test system vendor will help out. They supply calibrators and controls and reagents that they manufacture under highly regulated conditions. And so that provides a means of assessing measurement uncertainty during both qualification and during routine operations to monitor the performance. But understand that this whole system is designed not only for your use, it's designed to cover a broader use by many different clients. And so they may not be exactly what you need. And so you need to explore that, and also understand that they have suppliers that can change, and that what they're supplying to you may over time change and may require that you not only revalidate all of these calibrators and controls and reagents, but that you adjust what you report out to your client.
In our laboratory, we used to say that you could drive a truck through the vendor's ranges.
Yeah. So we're going to drill down here, and I'm going to just take a moment to bore you with something that I think is really important that will then become more evident in future talks, why we've drilled down on this. And that is that, when you measure and analyze a specimen, that employs any number of possible physiochemical or biochemical reactions. So you could be talking about a fluorescent tagged antibody that combines with a drug, and then the signal that is created by that when it's exposed to light or to a laser will then be captured by a sensor, or if it's by say mass spectrometry, you're looking at a completely different type of system. But you always have a signal generator, and then it's captured by one or more sensors. So with flow cytometry, you may have half a dozen sensors, which of course makes things quite complex when we're talking about maintaining accuracy and precision. So the strength of the captured signal, which is also known as amplitude, is quantified by an embedded computer processor, and that runs a custom designed software. Well, guess what? So you're going from an analog to digital. So there's an analog to digital converter, and depending on how good it is, there will be a degree of error. Either it will underestimate the amplitude, which is the area under the curve, or it will overestimate. So you already have an error there. And then the software itself may not be well designed, and there may be error there. And that's translated mathematically then into a unit of measure that's related to the particular type of analyte. So the milligrams per deciliter or international units, whatever it is that you're looking at. So the translation is achieved by the vendor or by the laboratory that's developing the test by generating a nomogram. And what this is that you have a known analyte in a set of concentrations that should yield a known set of signal strengths based on the research that they do. And so a nomogram to take an unknown signal from an unknown amount of analyte in a patient or in a research specimen is then compared to that nomogram, and then that's translated back into the from the signal strength into the units. And the problem here, of course, is that that nomogram is calculated, or it's analyzed under a range that's known as like a range of linearity. The nomogram is actually an equation. And so even if you're outside the range, you can still generate a result. The problem is you need to know that if it's outside that range, it may not be linear, and so it may not be reliable. It could be very inaccurate. And often people in the field who use this, for example, for PT-INR, often clinics try to run these tests and interpret them and don't realize that beyond a very narrow range, that the results of PT-INR are basically like throwing a dart at a target. So each step of the process has a degree of error that can affect either or both test accuracy and precision. For example, establishing the nomogram, as we've talked about, calibrating the test system. So when the vendor brings this system into the laboratory, gets it installed, they have to make sure that it's properly calibrated, that all of the parts and pieces are working properly. The gears, the sensors, the light source, all of that is within a certain range of brightness, and so they have a way of testing this. And it even includes how much of a volume is drawn up by the system. And there are many different technologies that are used to try to reduce the error. And by the way, the smaller the volume that's going to be drawn up, the harder it is to maintain accuracy and precision because of simple limitations in these physical systems. And then, of course, to testing the unknown analyte itself. There are all sorts of problems there that occur. And this doesn't take into account numerous other sources of error: pre-analytical phase, method of specimen collection, specimen volume, as I've just mentioned, preservation, and then pre-test processing, all of which require that the person involved, the technologist, the blood draw, everybody do this correctly and do it reliably and uniformly. So
experience shows that contribution to the total measurement uncertainty is probably small, but it's important for each laboratory to identify those areas of potential measurement uncertainty and make a decision on whether or not it can have an effect on your total uncertainty.
And so what that means is that you want to be able to rank them, and you could do this using something called failure mode and effect analysis or some other way of ranking the seriousness of this. And then that's where you decide how you're going to monitor. You can't monitor everything, obviously. You can't look at everything. But then you can decide what are your most important areas and steps to monitor, and you monitor those. And when there's a problem with them, you go in and you make the changes. So
think in terms of the difference between doing a phlebotomy on a vein and doing a finger stick and the quality of the sample, as an example.
Yes, absolutely. It's a different matrix. And so if your instrument isn't set up to measure the analyte in the particular matrix, you may be presenting the end user with a very unreliable result. That's a really excellent point. So key issues regarding metrology that the laboratorian should consider. So when you're going to choose the test system, you want to look at the following metrological characteristics. Okay, and these include but are not limited to. When I say that, it means on your own, in your particular situation, it will vary. There will be other things that will be important that I haven't stated here. So there are going to be limits to the physiochemical laws. Rather, the physical chemical laws are going to limit the performance of your test system. So if you have an antigen antibody system that you're using against, say, a drug, say benzodiazepine, you can easily get a lot of false positives for other substances that look kind of like it. So that's a screening test. It's very sensitive, overly sensitive in some cases. And so you're going to then confirm with, say, mass spectroscopy or high performance liquid chromatography, something else. But that system may not, may be so specific that it misses something that's important. So each one of these has its own metrological issues. So the design and function of the instrumentation used to execute the test, how much automation, and what goes into that automation. Literally, if the gear is made of metal, that could be great. If it's made of a soft plastic to save money, that can be a disaster, where these things wear down slowly, teeth wear down slowly, and it turns out that the movement of materials is hampered, and you have the wrong results. The same thing with the sensors in terms of their sensitivity, longevity, the potential for them to wear out. So you get a drift, and also in the light source itself, and even in the computer components. So there still is a company that produces hematologic instruments, and one of their models, I had enough experience so when I had a client that was having problems with accuracy of their results, the hematologic results, I could tell them what component was going wrong. They wouldn't believe it, but it turned out that component was constantly going wrong for all of the clients because the supplier had gone out of business, the third-party supplier had gone out of business for that particular component. And so the only way they could keep those things running was to take the old component out of the instrument, take it back to the manufacturing plant, refurbish it, and then install it in somebody else's system. And that was really pretty amazing. So the reliability of the nomogram, you really need to make sure that that's the correct nomogram. I've seen cases where an instrument is upgraded, but they forget to put the new nomogram in. I've seen secondhand instruments purchased that have been upgraded, and the person who installs it has the wrong nomogram. So you need to make sure that you test your instrument against a known standard that is reliable, whether it be a reference lab you split specimens to, or the instrument you already have in your lab that's been, has been performing well, and the performance qualification has done well. Then there's the reliability of vendor calibrators and controls, so we've already mentioned. And also the reagents. Those are things you have to look into. Then there are ancillary supplies, the quality of the pipettes and the equipment such as centrifuges. Deborah and I did a study in a research laboratory where they were using high performance liquid chromatography, and the receptacles into which very small amounts of specimen were being placed, the caps were no good. And it invalidated the results of the study because the caps had been replaced. One supplier had been replaced by another, and nobody had thought to validate that those new caps worked properly. That's how important this can be. That's where little things mean a lot. And then the comprehensiveness of the qualification process to characterize the test system. You want to have enough sample size so that you really are certain you have an accurate average, an accurate standard deviation, two biggies that are used to calculate all sorts of other things. For example, the CV, the coefficient of variance. They give you an indication, standard error, all these things. You want to make sure that the calculations are going to be accurate. So it's really important that it's comprehensive and that you've stratified and that you for the intended range of values that you're going to most normally be getting, that you have good linearity. Then the complexity and appropriateness of the procedures to operate the test system. If it's just too much for your laboratory, it doesn't matter how accurate and precise the instrument is, you're not going to be able to get those good results. And then the systems you put in place to monitor the performance. If it's high output, you may need to put a control at the beginning of a batch and at the end, because believe it or not, during the run, especially in things like high performance liquid chromatography, but also in large clinical analyzers, there can be enough drift so that the results you get at the end are not going to be in control. And you won't know it unless you run controls at the beginning and at the end. And then finally, training of those assigned to operate the system. The training has to be thorough and ongoing. And then the competence of those operating the test system, which also has to be thorough and ongoing, using proficiency testing through third parties, or you might have something internally that you do to make sure that they're not drifting in how they perform that procedure, which could affect the metrological characteristics. And you want to notice there's a potential issue here. Vendor provided material to qualify and monitor the reliability of the test system. It's because the vendor's perspective, they need to design the system to meet the requirements of a wide variety of potential customers. And so these might not be adequate for you if you have a really very important situation, and you need to make sure that what you're doing is going to meet the needs of your end user. So next, in conclusion, it's the careful study and attention to all these metrological underpinnings of the laboratory test systems, whether it's vendor provided or whether you develop it internally, and so that you can assure that you're doing the following. You choose the right test method and system for the mission. That you minimize the risk for deploying a test system that does not support that mission, where you go through the whole process and in the end user goes, this just doesn't work for us. We can't use these test results. And Deborah and I have both had this experience where a test system doesn't even put out test results in a manner that can be interpreted by the clinician, because the vendor has used different units that don't really match what the end user is used to.
Or worse, they interpreted incorrectly on their own.
Yeah. Maintaining reliability during routine operations to assure success of the mission, because the mission may be long-term. Obviously, for the clinician, it's forever. It's over years. But for for example, for a research organization with a drug development, for that particular drug, it may be 3 months, 6 months, whatever, a year. And then after that, they don't really need that test anymore. Now it's gone to manufacturing, and they have a whole different system of maintaining the reliability of the manufacturing. And this is achieved by basing your decisions on the end user stated acceptable measurement error. Okay, so we keep on coming back to this. Why? The reason why we come back to this is that you shouldn't be the ones determining what this error is. The end user, based on what they know and what they need, it should be them, because they need to decide, well, what's the acceptable risk to their patients, or in terms of the drug product going into the mouths of thousands or tens or even hundreds of thousands of people. What is acceptable for error here, in terms of not only research, but also for the manufacturing process, and also for getting your studies through the FDA if you're dealing with the FDA.
Go ahead. Sorry. No. I just like to say that this may sound to some people like it's a presentation for picking an analyzer or picking a system, but please remember that applies to systems that you have operating now that maybe you're not aware of all the areas of measurement uncertainty that you could be.
Yes, and especially with point of care testing, which is a really big area where accuracy and precision, if you've got, by the way, if you've got 20 point of care testing instruments throughout a hospital, not only should they be qualified, but they need to be qualified against each other, so you know how much difference there is between these instruments, and hopefully you can cluster the ones that are closer to each other in a particular ward, so that you don't test with one instrument and then you test with the other one. And by the way, suddenly the test results are, quite different from each other, and you think the patients had a change in their biologic or clinical status when in fact they have not.
I think that's probably a whole nother series of discussion.
Yeah. So in follow-up presentations, we're going to look at a number of concepts in more depth. And by the way, what we present, it can be also determined by you, the listener. If you have a specific need and you'd like to learn more, obviously we would like to provide that for you. So concepts like accuracy and precision, which are often not well understood. We think we understand them, but then when you get really deep into them, you realize that there are a lot of aspects to this, to the definitions that need to be assessed. Limits of detection of any particular methodology and the physical system used to implement that technology. Then the limits of quantitation in terms of being able to quantify something. Then the definitions of the following. Design qualification. So we're used to IQ, OQ, PQ, but the design qualification is also important. You need to determine, does this design of the instrument fit with a laboratory in terms of its operations, its personnel, and the needs of the end users? And then installation qualification, of course, operational, and then performance qualification. And then covering such things as, how these can impact the performance of the test system metrologically, to estimate them through appropriate sampling and statistical analysis. That's something that we can go over. How to report these to the end user in a way that helps them properly interpret test results. So just the way it's communicated, this error is communicated, it's very important that the end user fully understand what it is they're looking at, so that they don't misinterpret it and make a wrong move, even though you were giving them really important information. So now we'd like to open this up to questions.
You can submit your questions by using the Q&A or chat function at the bottom of your screen. And we did have a couple actually come through, Mark and Deborah, as you are presenting. I can start. One of them is, what do you mean by acceptable risk criteria, and who determines this?
Yeah, so back about 10 years ago, the Clinical Laboratory Standards Institute implemented something called EP23, which I highly recommend that you obtain that document. I got a chance to be involved with that. And the centerpiece of it is that the laboratory needs to use various methods, such as looking at their control data, to determine what the variation is over time for the particular laboratory test result that they're generating, and to report this out to the clinician. But they also felt that it was important for the clinician, or even the risk manager for a hospital or for a clinic, be involved with determining what is acceptable in terms of the variation of the laboratory results. And so and also this can be done by many means. So in one case, you may have a gold standard against which you're looking at in terms of a test. So you might have the gold standard in the laboratory, it's your laboratory instrument, and then you have point of care testing instruments. So what is the concordance or discordance in terms of the final diagnosis made if you take the same specimen and you run it on both instruments? So if the laboratory instrument gives you, out of say 20 patients, that 5 are positive, and that means that a diagnosis is made for this particular physiologic state or actual illness, and the rest are negative. Well, what happens if the point of care testing, there are 7 that are positive and 13 that are negative, or there are only 2 that are positive and 18 that are negative? It means that there's discordance, and you can calculate the percentage of discordance and ask the question, is this acceptable? Is it acceptable for patient safety, for the liability of the hospital? These are and for the liability of the physicians using it. These are the questions that need to be answered. Next question.
Thank you, Mark. The next question is, what does the lab do about the other sources of error not talked about?
Do you want to take that, Deborah?
Sure. So as we said, we listed all of those pre-analytic potential sources of error. Typically, what you do is you identify all of them. And as Mark said, you rank them to see what the not only what the variation in error that it can occur, like the example I gave between the phlebotomy and the finger stick, obviously folks that are doing those kinds of tests in the laboratory know there's a variation between a phlebotomy and a finger stick. And then you determine whether or not that has an impact on your total, a significant impact, essentially. You rank them. And most of those do not end up having an impact compared to all of the other measurement types of uncertainty that you can have. But there may be something like the example I gave where you want to consider that when you're measuring your total uncertainty and move that particular task up in the ranking. So the important thing that I would want to look at when I asked you, how did you determine your measurement uncertainty? I would like to see that you have identified all of those, evaluated them, and made a risk assessment about them. But you may not have to do anything else with them.
Jen, I noticed someone has their hand raised. If you would type your question or comment into the chat, we can take your question.
All right, thank you, Deborah. We do have another question, and this is, what do you suggest we do about vendor supplied ranges for considering the test results as good?
Excellent. I think both of us will answer in part. So it's okay at the beginning when you haven't collected a lot of data. Once you run your controls for several months, you're going to be able to see, how tight is the variation from the ranges that they've given. Either it goes well beyond their ranges and you've got a problem because it's constantly out of control, or you find that it's a lot tighter, and you can look and see whether or not that should really be your range. That if it goes beyond that, even though it's below the range that has been given by the vendor, if it goes beyond a range that you can establish statistically, then that's probably when you should be looking into a potential source of metrological error.
And you know what, Mark, excuse me, but I had that exact problem with a benchtop whole blood alcohol level. And we had a tighter range and a tighter precision than the vendor. And when we started having problems with the analyzer, we called the vendor and they said, well, it's within our range, so it's fine. And I said, no, but our imprecision has gone from 3% to 5%, which is still within your range, but we're having problems with our controls and we've gone outside of our qualified range. And I can tell you that it was quite a debate with the vendor to send a field service person in, and he got into the back of the analyzer, the part that we couldn't get into, and he found that the stepping motor belt was frayed. So it randomly caught and didn't, as Mark said, the pipetting of the sample varied just whenever that thing caught. So that's an example of where our qualified range informed us that there was a problem and we were still within the vendor's published range.
Yeah, so you might want to call us gearheads, but we've had this experience where the gear makes all the difference. Yeah. But yeah, and so it's really important to be able to do this. Next question.
All right. I believe we have time for one more before we'll be wrapping up today's presentation, but they're asking, can you explain what the lab does to give a clinician an idea about the testing variation?
Yeah, so another life experience. I had a doctor get very angry at a technologist over quantitative beta HCG. And as I entered the scene, I heard him say, I almost took this patient to surgery because of your result. And what we found out was that he had come from a different hospital that was doing quantitative beta HCGs by radioimmunoassay. And to him, a 5,000 milli-international unit change in the patient over 48 hours was significant for his diagnosis and taking her to surgery. In our method, we were measuring kind of, our mean was around 30,000 milli-international units. And day to day, we would have a 5,000 variation in that number on the same sample. So he was quite horrified to hear that. And as a result of that, we set up a plan to pair specimens. So they told us to hold the specimens. They were trying to rule out ectopic pregnancy. And then they'd sent the second specimen 24 or 48 hours later, and they would tell us to run it with the first one. We paired them on the same run, and we minimized the test variation so that we could tease out the variation in the patient. And then I gave a presentation to all the OB doctors about that process. Now, every test is not going to be that extreme, but those are the kinds of things you need to understand their decision levels, as Mark said, their acceptable risk, so that you can make sure that they understand the limitations of your procedure.
Yeah, and I think the really key term here is to educate the end user. That's really important. And in the VA, that's what really was happening, was we began to educate them on hemoglobin A1C. And then they could not only see that here, if the result varies this amount from the original result, plus or minus, then it's probably laboratory variation and not the variation in the patient's clinical status. So don't up their dosage of metformin because then they can become hypoglycemic. That's more dangerous than being hyperglycemic for a little while.
I think a good common example is we've all seen the commercials for oatmeal, and the person says, I lowered my cholesterol by 9, whatever, they don't even give units. Well, if your cholesterol is 340 and you've lowered it by 9, that's probably run variation and not really a change in the patient that's medically significant.
And it means that the tendency to want to call something abnormal, for example, it used to be 146 milligrams per deciliter for glucose was considered abnormal. Then it was dropped to 26. There's some in the field who want to drop it to 106. Well, the metrological characteristics of the instruments won't work. You'll end up calling everybody at some point diabetic or pre-diabetic if you were to ratchet it down like that. So sometimes it's really the educational aspect of it to let the clinicians know the technology simply can't get you there. So I think we're done.
Yeah, thank you, Deborah and Mark, for this presentation, and to our attendees who have joined us. We will be sending out a link to this webinar to you guys, and it will be also available on our website at labshire.com. Please stay tuned. Like Mark had mentioned before, this is going to be a series of webinars, so stay tuned for future invites that we'll be sending out. And I'll just kind of pass things over to Dr. Blackwell to wrap this up.
Oh, thanks. Can you put my slides back up? Thanks.
Oh, sorry.
That's all right, Mark.
I didn't. Can you see it?
Not yet.
Oh, wait a minute. Okay, sorry. But it's going to take me a moment here to.
Yeah, well, Mark's pulling that up. Thank you for attending. We can help out with many aspects of your laboratory, really from site selection all the way through late life cycle issues in terms of your laboratory. So if you have any questions, reach out using the contact information here. We'd be happy to talk to you. This is an overview. Mark alluded to this, or these are some general topics and areas that we'll be talking about in the future. We just talked about one aspect of accuracy and precision in the first one, but there's many important aspects to laboratories that people need to be aware of and can possibly need consulting help with that. Thank you.
All right, well, thank you. This concludes our webinar for today. If you have any questions, please feel free to reach out to us at info@labshire.com. Have a great day.
Bye.
Bye-bye.
Bye, everybody.